Greetings, everyone, and thank you for joining us today for the 2023 Henry Pickering Bowditch Award Lectureship webinar. My name is Patricia Terrell with the American Physiological Society, and on behalf of APS, I would like to thank our lecturer, the organizers, and moderators for your support. Before we begin, please allow me to present a few housekeeping notes. The webinar is being recorded and will be made available for on-demand viewing. To maintain scientific integrity and respect towards our award lecturer, please refrain from photo or video capture of the scientific presentation in whole or in part. Now, during the lecture, please post your questions and comments via the Q&A feature instead of the chat. You may post them at any time during the lecture, and they will be read at the conclusion. It is now my pleasure and honor to introduce our distinguished moderators, Dr. Dee Silverthorne, the current and 95th president of APS. She is also a distinguished teaching professor of physiology emerita in the Dell Medical School at the University of Texas at Austin. And joining Dr. Silverthorne is Dr. Willis Rick Sampson, the 96th president-elect of APS. He is also a professor of pharmacology and physiology and the director of biomedical science graduate programs in the School of Medicine at St. Louis University. Well, I'm sure that you are anxious to begin, so I will now turn the webinar over to our moderators, who will introduce our Henry Pickering Bowditch Award recipient, Dr. Gina L.C. Yosten. All yours.
Thanks. Hello, everyone, and welcome to our annual Bowditch Award Lecture. I'm Dee Silverthorne, current president of the APS, and I'm joined by Rick Sampson, who is our president-elect. The Bowditch Award Lecture is given annually by the American Physiological Society and is one of our top honors. It is named for Henry Pickering Bowditch, who was the first president of the APS. Dr. Bowditch received his medical degree from Harvard, but instead of going into clinical practice, he chose to go to Europe and study physiology, first with Claude Bernard in France, and then with Carl Ludwig in Germany. He returned to Harvard as a faculty member and eventually became the chair of the Department of Physiology there, where among his distinguished proteges was another famous physiologist, Walter B. Cannon. In 1887, Dr. Bowditch, along with 4 colleagues, founded what became the American Physiological Society, and he served as the first president. In 1956, the Society started the Bowditch Award Lecture in his honor. It is presented every year to a young physiologist no more than 42 years old or who is within 8 years of starting their first position after postdoctoral training. And it recognizes outstanding and original accomplishments in the field of physiology. So to introduce this year's Bowditch awardee, I will now turn the microphone over to Rick Sampson.
Thank you very much, Dee. Thank you, Dee. I hope I can get my screen up. I must be doing something different then. Speaker view, okay. Does mine come up? I guess not. That's all right. It's my distinct pleasure to introduce this year's awardee, Dr. Gina Yosten. Gina grew up on a dairy farm in Munster, Texas, and from an early age was interested in science and physiology. She completed her bachelor's degree and was a Phi Beta Kappa at the University of Oklahoma, and then spent 2 years at the Oklahoma Medical Research Foundation examining her future career goals. It was my great fortune that in 2006, she joined the graduate programs here at the School of Medicine at St. Louis University and completed her PhD in pharmacology and physiology in 2010. Gina stayed on as a fellow here at SLU and did a brief fellowship with Jane Reckelhoff at University of Mississippi, and then was appointed as an assistant research professor in 2013. She was elevated to the tenure track in 2015, and in 2020 was awarded tenure and promoted to associate professor. This was a big year for Gina. She also became the editor-in-chief of AJP Regu during that period of time. As you can see, Gina has been very successful, not only in her research, but acquiring the funds to do that research and to build a very large group here at SLU. Now, Gina has many achievements, and what most people don't know is that her first publication actually was awarded the top poem of the year at the Cowboy Poetry Gatherings. So it's appropriate that we show that she's a cowgirl from the very beginning. Gina has several hallmark achievements in the laboratory. While a graduate student, Gina was an integral part of our discovery of the hormone neuronostatin, which is part of the somatostatin prohormone. And she has gone on to characterize the physiologic relevance of neuronostatin and its potential therapeutic value in the treatment of diabetes. While a postdoc, she was the discoverer of a second previously unrecognized neurohormone, phenixin, which she continues to work on. But actually, the hallmark of her work is the fact that she developed, and she will talk about today to a certain degree, a strategy for de-orphanizing previously orphaned G protein coupled receptors. And currently, there's 5 on the list, and I can tell you that there's probably more on the way. Gina also has 2 patents related to those de-orphanizations that she was responsible for. And those patents are addressing the therapeutic value of identifying these receptors and developing small molecule ligands for therapeutic use. During all this scientific discovery and these activities and travels throughout the world lecturing on her work, Gina actually has an even greater accomplishment, and that is the 3 daughters that you see here in this picture: Gwenny, Kenzie, and Maddie. So with no further ado, I'll turn this over to Gina for the 2003 Bowditch Award Lecture.
Well, thank you very much, Rick, for that really great introduction. And thank you especially to the selection committee for choosing me for the 2023 Bowditch Award. Professor Sampson sent me the list of previous awardees for the Bowditch Award, and it's actually truly humbling to be able to add my name to that list, which includes Nobel laureates, our Cannon Award lecturer for this year, Kurt Sigmund. Hopefully, however, well, I think I might be the only cowgirl poet on that list. So perhaps I don't have to feel like such a dwarf compared to the giants that are already there. So as Rick mentioned, over the last few years or so, last decade or so, my group has been very interested in the role of G protein coupled receptors in physiology. And in particular, we are very interested in orphan GPCRs and figuring out what the ligands of these orphans are, and then what they do as a ligand receptor pair. And I'm going to tell you the story of one of these receptors that we've been working on, really focusing on over the last 5 years or so, that sits at this intersection point between organ systems. But before I tell you about that receptor, I would like to start by telling you about a patient. So our patient was a 51-year-old woman who discovered a large mass in the right upper outer quadrant of her right breast. She presented to her primary care physician, who immediately sent her to radiology for a mammogram. Mammography revealed a very large radiologically dense tumor, so 7 centimeters. And just to get a visual of how big this tumor is, it is the size of my fist. There were prolific calcifications associated with this tumor, so the radiologist graded it as a BI-RADS 5, which means highly suspicious for cancer. Now, the radiologist then sent our patient to have a needle biopsy, and the pathology came back as a triple negative ductal carcinoma, grade 4. And in addition, there was partial lymph node involvement. So the final diagnosis of our patient was ductal carcinoma, triple negative, grade 4, stage 2. Now, for those of you who are not necessarily really into the breast cancer field, this triple negative grade 4 cancer, this is the bad breast cancer. This is the cancer that the type of breast cancer that is usually very resistant to chemotherapy. So this was a very grave diagnosis for our patient. So our patient underwent the standard treatment course for this type of cancer, which included 8 rounds of paclitaxel and doxorubicin chemotherapy. And then, fortunately, unlike many triple negative cancers, our patient's tumor shrunk from 7 centimeters, the size of my fist, down to a smaller tumor that was 2 centimeters, the size of my thumb. The physicians performed a lumpectomy of the remaining tumor and then performed targeted radiotherapy to clear out any remaining tumor cells. Now, during the course of the treatment, the patient reported a drastic loss of appetite and really significant weight loss. So she lost over 30 pounds during treatment. And in addition, she developed during her last doxorubicin treatment, painful peripheral neuropathy of both of her feet. Now, this patient is a real patient. Her name is Linda. I usually just call her Mom, and you can see her here with my youngest daughter, Gwenny. And we were very fortunate because the treatment worked. She has been cancer-free for 15 years now. And the anorexia associated with her treatment has resolved, but she still has sustained painful peripheral neuropathy of both of her feet. And this pain has been unresponsive to opioid therapy and unresponsive to even experimental interventions. And as it turns out, my mother's experience with the side effects of treatment and the physiologic effects of having cancer are not all that uncommon, because we know that up to 85% of cancer patients have some form of cancer-associated anorexia. And while the etiology of this anorexia is pretty diverse, for instance, it can be due to the disease itself, a hormone secreted by the tumor, or from the treatment for the cancer. But regardless of the etiology, it appears to be due to changes in the hypothalamic and brainstem feeding centers in brain. And this is really important because we know that severe anorexia and weight loss, which can or might not be associated with a cachexia phenomenon, is usually associated with a much poorer prognosis in many types of cancers, including breast cancer. And in addition, my mother's experience of painful chemotherapy-induced neuropathy is not uncommon either. So in this study published in Pain almost 10 years ago, and this hasn't changed much, of breast cancer patients 1 month following cessation of treatment, you'll see that 70% of these women experienced painful peripheral neuropathy. Now, while there was a slight resolution at 3 months to 60% who experienced peripheral neuropathy, at least a third of breast cancer patients who undergo chemotherapy treatment have sustained peripheral neuropathy that lasts longer than 6 months. In my mother's case, it has lasted for over 15 years. So clearly there is a need to be able to attack both of these issues that are associated with cancer, but are not treated with chemotherapy. So the anorexia as well as the pain. Now, the story I'm going to tell you starts as many stories do, and that's with a poster session. So I was actually thinking of naming this talk today, Always Go to the Poster Sessions, because it's really true. You get really good conversations at poster sessions, like the one that this poster was presented at the Physiology Summit when it was still called Experimental Biology. Now, this poster was talking about work that really predates our receptomics strategies that I'm going to talk to you about today. It was really a precursor to our receptomics that we presented at Experimental Biology, which is now Physiology Summit, and then was hanging in the hallway outside of our door. And my colleague here in the department, Daniela Salvemini, happened to see this poster. And she is an expert in chronic pain mechanisms. And she asked me, so Gina, do you think we could apply some of this strategy to identify GPCRs that are involved in different types of human disease? Could we apply this to my model systems? And of course, we said, sure. So together with Daniela, we developed what is now our typical receptomics strategy, which is based on bioinformatics. So the way that this algorithm works is we start with a geodatasets query. And in this case, we were looking for GPCRs that were significantly changed in the setting of chronic constriction injury, which is a rodent model of neuropathic pain. And we looked in the spinal cord to see which of these GPCRs was upregulated. You can see that quite a few were upregulated. So we applied phylogenetic analysis using the Grisham tree method to cluster these receptors into families. And when we took a closer look at this tree analysis, we found that there are basically 4 major branch points of our phylogenetic tree. We then took those branch points and looked at the homologous regions of all the members of those branch points. And you can see that we've named them here: opioid-like, purinergic-like, serotonin, cannabinoid, and arachidonic acid-like. And we performed a blast screen using the homologous regions of these different family members to look for orphan GPCRs that were associated with these families. And quite a few popped up. But when we went and did a preliminary screen in spinal cord of the rat to see which of these receptors was actually present in the spinal cord, we found that just the GPCRs that are highlighted here in bold or bolded here were actually expressed in the spinal cord. We could then take those receptors that we knew were expressed in the spinal cord and performed a functional qPCR-based expression assay to see if there were any functional changes in expression in the setting of neuropathic pain. So we took some of Daniela's rats that had undergone chronic constriction injury, and we took out their spinal cords and looked at expression of these different receptors in the dorsal horn versus the ventral horn of the spinal cord on the ipsilateral side, compared to the side that was contralateral to the site of injury. And what we saw right away is that there were 3 receptors that popped up, with GPR 160 showing the greatest increase in expression in the setting of chronic constriction injury-induced neuropathic pain. So we went on to see if the protein was actually expressed in the rat spinal cord as well, and it was. and it's expressed right where it should be if it's mediating effects of neuropathic pain, and that's in L1, L2. And through the collaboration with our good friend Grant Kohler, we then performed RNA-scope analysis to see what would happen to expression of GPR160 in the spinal cord in the setting of neuropathic pain. So in a CCI, chronic constriction injury model, we found that on the ipsilateral side of the spinal cord, we saw this significant accumulation of GPR160 in the dorsal horn, suggesting that GPR160 may in fact be involved in the transmission of pain. So to test whether GPR160 is involved in mediating the pain signal, Daniela's group used an siRNA against GPR160 and found that in her CCI model, whenever you treat animals with GPR160 intrathecally, that it completely prevents the development of neuropathic pain, which is read here as a Paul withdrawal threshold. So a lower number means higher pain. Not only that, but Daniela was able to show that if she induced neuropathic pain and then treated with siRNA against GPR160, she could reverse the neuropathic pain that developed prior to treatment. So this was very good evidence that, yes, GPR160 may be involved in transmitting the signal that mediates neuropathic pain. And that's all fine and good. And it could be that GPR160 would be a good therapeutic target for neuropathic pain. And we were really interested in whether it could be a therapeutic target in chemotherapy-induced neuropathic pain. But really, in order to answer that question, we needed to know what the ligand of GPR160 was. And so to address this question, we applied a deductive approach. So we first went to the Human Protein Atlas to see where it was known that GPR160 was expressed in what organ systems. And there were quite a few that popped up. But when we compared the expression profile of GPR160 to the expression profile of multiple orphan ligands, these are hormones, peptide hormones, or other types of ligands that we know exist and have biological activity, but we don't know what the receptor is. And we compared their expression profile of these known orphan ligands to the expression profile of GPR160. And really only one of these orphan ligands was expressed in all of the tissues where GPR160 was expressed. And that was cocaine and amphetamine regulated transcript, or CART. Now, CART is a peptide hormone and neurotransmitter that was discovered in 1995 by Douglas and colleagues, who were looking for transcripts that were upregulated in rat striatum following treatment with cocaine or amphetamine, hence the name cocaine and amphetamine regulated transcript. And although CART is known to be involved in reward systems, it's also now known to play a role in multiple physiological processes, and including appetite and pain. So, for instance, it's known that in rats, when we and others have shown that CART injected into the fourth ventricle reduces food intake down to almost nothing. And in addition, when you knock out CART in mice, you actually get an exaggeration of body weight, which could mean that CART is a physiologically relevant regulator of appetite and of body weight. And this appears to hold for humans as well, since it's known that single nucleotide polymorphisms in the human CART gene are associated with human obesity. And in addition, we know that CART is present in the C fibers of the spinal cord and elicits signaling in the spinal cord neurons via phosphorylated ERK. And in addition, injection of CART has been shown to lead to a painful response. So it would seem that CART could be a good candidate for the ligand for GPR160. So to answer the question of whether there was a connection between CART and GPR160, we applied part of our deductive reasoning strategy that we've used to deorphanize. We, at this point, had used to deorphanize 4 other orphan G protein coupled receptors. And the way that the second half of this process works is we take cells that we know are responsive to our peptide of interest. We use siRNA to knock down our GPCRs that we think might be involved in mediating their signal, and come back and treat those cells with the peptide and see if we lose responsiveness of these cells to that peptide. So in this case, we were using a cell line that we use a lot, which are the Cato3 cells. This is a human gastric carcinoma cell line. Is it a physiologically relevant cell model for this project? Not really, but it does have GPR160 highly expressed, and these cells will respond to treatment with siRNA very well, which you can see here. We got a significant reduction in GPR160 expression when we applied GPR160 siRNA. Now, when we did this, we found that these Cato cells, which normally respond to treatment with CART with a significant increase in CFOS mRNA expression, which is determined by qPCR. However, whenever we pre-treat these cells with siRNA against GPR160, we see that we completely lose this effect of CART on CFOS. And this effect of CART is not just due to something weird that the siRNA is doing, because the control siRNA does not block the effect. So this is our first evidence, really, that there was this functional relationship between CART and GPR160. But again, Cato3 cells are a cancer cell line. They are not really a physiologically relevant cell model for assessing the effect of CART in cells and whether or not CART and GPR160 are a ligand receptor pair. So to investigate this more fully, we looked into a more physiologically relevant cell line, and that is the PC12 cells. Now, PC12 cells are a pheochromocytoma cell line that can be differentiated into a neuronal cell-like cell line with treatment with nerve growth factor. So we took PC12s and differentiated them into these neuron-like cells, and then treated them with CART. But before we did that, we had to know whether or not our receptor was actually there. So in PC12 cells that have been differentiated, we found that GPR160 was highly expressed, as was our 2 other receptors that I showed you in that initial qPCR screen were highly upregulated in the spinal cord following induction of pain, and that's GPR18 and GPR183. So knowing that GPR160 was there, we figured that if 160 was in fact a receptor for CART, that CART treatment in the differentiated PC12 cells would cause a change in signaling. And we knew from the literature that CART is known in other cell types, and including in PC12 cells, to induce phosphorylated ERK signaling. And that's exactly what we saw. So when cells were treated with CART, we saw this nice induction of phosphorylated ERK, which you can see quantified here on the right. But that doesn't, of course, answer the question of whether this effect is being mediated by GPR160. So we went back and treated some cells, again, with our siRNA to GPR160, and found that we completely lost the effect of CART on upregulation of phosphorylated ERK whenever GPR160 was significantly knocked down. So this is, again, further evidence that GPR160 and CART are functionally interacting. But that doesn't answer the all-important question when you're trying to determine if a ligand and a receptor are a ligand-receptor pair. And that's the question of whether there is a physical interaction between CART and GPR160, in addition to that functional interaction. So to approach that question, we used a few different methods. So we started with a co-immunoprecipitation assay. So we took our PC12 cells and we treated them with CART, and then we performed an immunoprecipitation for either GPR160 or GPR182 as a control, and then performed a Western blot using antibodies against CART. You can very clearly see this, this band that shows GPR160 and CART together, which is not present in the control lane. Now, in addition, we took our CATO3 cells again and looked to see if there was any co-localization between CART and GPR160 on the cell membranes of these cells. And you can see that these cells, which were treated with CART, shown in the red, co-localized very nicely with GPR160 shown in the green, so that the merge is in yellow. Now, this is all fine and good. However, even with deconvolution algorithms at the time, so this was a few years ago, the best resolution that we can get with this experiment is about 200 nanometers. And we really wanted to get a higher resolution to be able to say with more confidence that CART and GPR160 were physically interacting. Now, to do this, we turn to the proximity ligation assay, which is an assay in which antibodies against your two targets are conjugated to essentially PCR primers. And if they are in close enough proximity, those two halves of the oligo will interact and initiate a rolling circle amplification that shows up under the microscope as these dots, these fluorescent dots. So that anywhere you see a dot, these two antibodies, in this case against CART and GPR160, are within 20 nanometers of each other. So a greater than a tenfold increase in resolution from co-localization. So you can see in our untreated cells, these are cells that were not treated with CART, you can see that there is some background, which we do see with the proximity ligation assay. But after 5 minutes, and especially after 10 minutes of treatment with CART, you can see a significant increase in the number of these puncta that indicate co-localization or less than 20 nanometer association between CART and GPR160. And all this taken together, we conclude that GPR160 and CART likely are interacting physically as well as functionally. Now, the important questions then became of whether GPR160, since we now think that GPR160 is at least a receptor for cocaine and amphetamine regulated transcript, was whether GPR160 is important for mediating some of these known effects of CART, keeping in mind that we're targeting people who have been treated for cancer and are experiencing both anorexia as well as neuropathic pain. So the first question that we wanted to ask was whether GPR160 was important for mediating CART's effects on food intake. So we started in rats that were fitted with indwelling fourth ventricular cannulas. And we started with just a neutralizing antibody against GPR160 and treated those animals with this antibody prior to the injection of CART. Now you can see in the gray bars, whenever you have CART treatment, that you get this reduction in food intake, which has been shown many times by many people. But whenever you pre-treat the animals with this anti-GPR160 antibody, you completely reverse the effect of CART on appetite. Now, with passive immunoneutralization, you can get all sorts of difficulties because the receptor is still, in fact, there. It's just hopefully being neutralized. So we also decided to take the approach of siRNA and used our standard protocol for siRNA-mediated knockdown in rats, which includes 2 consecutive injections of GPR1s of siRNA over the course of 2 days. And whenever we were able to knock down GPR160 significantly in the brainstem of rats, we found that this was associated with a complete reversal of CART's effects on food intake. So it does appear that GPR160 mediates the effects of CART on appetite. But then on the other side of the coin, is GPR160 involved in mediating the effects of CART in neuropathic pain? So we know from Daniela's work and from other people's work as well, that whenever you inject CART peptide intrathecally into rats, you get these reductions in paw withdrawal threshold, which indicate mechanoallodynia, or in other words, the induction of pain. So to see if blockade of GPR160 could reverse this effect, Daniela's group used the passive immunoneutralization technique that we had used for the food intake, and found that pretreatment intrathecally of this antibody against GPR160 completely reversed the effect of CART on neuropathic pain. This effect was also blocked by pretreatment with a CART antibody, as well as an inhibitor of the phosphorylated ERK MAP kinase pathway. So it appears that GPR160 is mediating the effects of CART on both appetite and on neuropathic pain, which could indicate that GPR160 and CART would be good therapeutic targets in the setting of, for instance, breast cancer, in which patients are experiencing high levels of anorexia, as well as neuropathic pain due to the chemotherapy. But the one thing that could derail this entire approach for treating these 2 ailments associated with cancer are whether or not CART and GPR160 are tumorigenic, since we wouldn't want to give a cancer patient something that is going to enhance the growth or metastasis of their cancers. Now, we have started, and Daniela has started to investigate the role of CART and GPR160 together in cancer progression. But there was some evidence in the literature when we first started on this project, looking at CART and GPR160 separately in different types of cancer. So for instance, in this study, it was shown that CART expression in breast cancer can be prognostic. So people who have lower CART levels in their tumor have higher overall survival in the total cohort and in the lymph node negative cohort as well. So having high CART expression in these tumors indicates a poor prognosis. Now, separately, another group was investigating GPR160 expression in prostate cancer and found that as progression of prostate cancer was increased, we also saw increased GPR160 expression. So the later the stage of the disease, the higher the expression of GPR160. And many cell lines of prostate cancer, human cell lines, also express GPR160 as well. And these authors also found that whenever they took some of these cell lines that express GPR160, these prostate cancer cell lines, and treated them with a short hairpin GPR160 shRNA, that knockdown of GPR160 was associated with slower growth and higher death in these cell lines. So this would indicate that CART and GPR160 are themselves tumorogenic, which could indicate that an antagonist for GPR160 could be a useful therapeutic in cancer. So just to summarize what I've told you today, we believe that CART is a ligand for GPR160 due to the functional and physical data that we have regarding CART signaling through GPR160. We think that GPR160 mediates the anorexigenic effects of CART, and it appears that CART and GPR160 are interacting to contribute to the development of neuropathic pain. It also appears that CART and GPR160 signaling may promote tumor progression in at least 2 different types of cancer, including breast and prostate, but we are looking more into this potential effect. So from all of this, we conclude that GPR160 based antagonists could be very useful in the setting of cancer for chemotherapy-induced neuropathic pain, for ameliorating cancer and chemotherapy-induced anorexias, and even could serve as an adjuvant for anti-tumor therapy. And we are very hopeful that antagonists for GPR160 will serve as this, well, basically as a trifecta to help people like my mother and many, many other women who have experienced all of these deleterious effects of cancer, not only the cancer itself, but of the cancer treatment. So the most important slide of any presentation is this slide right here, which is the acknowledgements. And I have many people to acknowledge. I, of course, need to acknowledge my co-investigators, Rick Sampson, who introduced me, who has been a tremendous support and mentor for 17 years. I absolutely could not have done anything that I've done without his guidance. Grant Kohler, who has been collaborating with me for a very, very long time. We have collaborated even on 3 human synthesis projects, although to be fair, I did most of the work for those, for the synthesis of those humans. Grant has been, of course, an integral part in my career and in the group as well. I'd also like to acknowledge our most recent addition to the group, Joshua Stafford, who's a junior faculty member in our group and is great at keeping us all in line. I, of course, need to, I have many really great collaborators, but for this presentation, I especially need to highlight Daniela Salvamini, who is now the chair of pharmacology and physiology, and has been just a tremendous support of my career, and also has been a real joy to work with on this project. And Alicia Pate, who has been helping us a lot with a lot of the animal work. I really need to highlight one of our past trainees, Chris Haddock, who did most of the signaling work for this project, as well as our current dream team here at St. Louis University, our students. I am very lucky to be able to have such talented and intelligent students working with me. My current dream team includes those pictured here: Lisa Haganah, Amar Mahmood, Colleen Baki, Tara Steffen, Nilu Mirian, our most recent addition, Megan Pater, and our lab manager, Teresa Ennis. Of course, I also need to acknowledge those who give us the money to do what we do, including 3 institutes in the National Institutes of Health, the Helmsley Charitable Trust, and the Foundation for Prader-Willi Research. And with that, I want to thank the selection committee again for this honor, and I am happy to take any questions.
What a fascinating talk that was, Gina. I know Rick has heard this before, but I think it's got a lot of implications. We have the first question from Christina Nadaluti. She says, impressive and terrific talk. What is the relationship between pain induction and appetite metabolism? Do you think that the immune system and or the inflammatory response may be a missing link? Do you also think that it would be possible to modulate the diet to control CART and GPR 160?
Those are all very good questions. I'll start with the first one. And that is, what's the relationship between pain induction and appetite? And the answer is that it's a very complicated relationship. And it's hard to tease it out. So in some individuals, it could be that the pain itself is causing anorexia. We know that whenever you have chronic pain, people don't feel good enough to eat. But it could also be that the fibers that are transmitting the signal for pain are also transmitting the signals for appetite as well. And that's something that we're looking into. Could the immune system or inflammatory response provide a missing link? Absolutely, it could. We know that GPR 160 is expressed in microglia and have some evidence that it's expressed in monocytes as well. So there's definitely a possibility of an inflammatory component to this entire story. And again, those studies are ongoing as we speak. And then lastly, would it be possible to modulate the diet to control CART and GPR 160? So we know that there are changes in CART and GPR 160 expression with fasting and refeeding. We have not looked to see if there are specific nutrients that can control expression of either one or both of this ligand receptor pair. But it's worth looking into.
Okay, Gina, William Pierce would like to know, do you think CART is the only ligand for GPR 160? Might GPR 160 be activated by biased ligands? And is the intracellular coupling of GPR 160 variable?
I love this question. So is CART the only ligand for GPR 160? I have no idea. We don't have any evidence that there are other ligands at this point, but that doesn't mean that there aren't. There is always room for more things to be going on than we know about. So could it be activated by biased ligands? Absolutely, it could. We have not yet, I think, fully discovered what all of the interacting proteins are for GPR 160 or for a lot of GPCRs actually. It's very possible that there are receptor associated modulating proteins or ramps that associate with GPR 160 that could change its specificity for other ligands. And it could also of course be impacting downstream signaling as well. So the one thing that we know about GPR 160 signaling is that it's consistently, you'll see an activation of the MAP kinase pathway. Could there be other signaling pathways involved? Absolutely. We're fairly certain that GPR 160 couples to GIO, but it's possible that under certain physiologic circumstances that other G proteins are going to associate as well. And actually, I would expect it to. but we just don't have evidence for which ones it is yet.
So we have a question from Guillermo Romero, who says, excellent presentation. What do you know about the intracellular signaling pathways associated to GPR 160?
Right. So just as I mentioned, we do know that we see a consistent activation of phosphorylated ERK signaling pathways, MAP kinase, and we think that GPR 160 associates with at least G alpha IO, but there's always room for more things to be going on.
Okay, Drew Theobald says, terrific presentation. I really enjoyed it. Regarding your PLA data, do you know why the no treatment group showed signal? If so, how do you know the signal in the treated group is specific and accurate?
Right. So with PLAs, you always see some amount of background, but the problem we had with this specific assay is that we found out after we did it that the cells that we were using, that we were treating with CART and then performing the PLA, they actually produce CART themselves. So they were releasing CART on their own, and then we were detecting the interaction of endogenous CART with GPR 160, but that was then enhanced following treatment with exogenous CART. So it really puzzled us too. It was actually very annoying because we're like, why can't we get this stupid PLA to work? It's always showing really high background. And then we realized it was the endogenous CART that was giving us that high background.
So Samara Resk says, excellent presentation. Does GPR 160 express peripherally and play a role in peripheral neuropathy through CART, or is it only centrally specific?
Yeah, so we think it's peripherally expressed as well. We don't have a full complement of where everywhere in the body that GPR 160 is expressed, the protein. So others have looked at message, but as we all know, message doesn't always translate into protein. Haha, that was a good pun there. Wasn't even intended. And so we haven't done a detailed analysis of GPR 160 protein expression.
And that I had a question that sort of follows off of that, which is, do you have any idea if this is in any way related to the peripheral neuropathy that might occur in diabetes? Because that would be a huge thing in terms of treatment.
Absolutely. And we actually do think that it might be playing a role. And we have some studies planned with Daniela Salvemini to address that question. And it's something that we're very interested in as well, because we focus largely on metabolic diseases like obesity and type 2 diabetes. So it's something we're very interested in.
So we have two more brief questions that have come in. The first one from Aichu Adera Gitu. Thank you for an excellent talk. Did you check that GPR 160 pull down CART and vice versa?
Yes, we did.
And Hannah Al-Zamil says, interesting topic is when there is a low level of GPR 160, is it correlated with obesity?
Right. So in rodents, so we're just now starting to be able to address this question. So we've developed a floxed rat model where the coding exon of GPR 160 is floxed, is flanked by LOXP sites. And so we can start to knock it out specifically in, for instance, different regions of the brain that are involved in appetite. And we're starting to do that now and starting to analyze the data. In humans, we don't know. So one of the great things about orphan GPCRs is that there's not a lot known. So there's a lot you can do. But the really bad thing about working with orphan GPCRs is that there's not a lot known. So you don't have a lot of good background information to rely on. So there's just not a lot of information in humans of whether there's any SNPs, for instance, in GPR 160 that might be associated with obesity syndromes.
We have a question from a past president of APS. Hello, David. David Pollock. Congrats, Gina. Do you know anything about this in sickle cell pain and or nociceptive pain?
In sickle cell pain, I do not, but that's really a very interesting question. So it appears to be involved kind of broadly in a lot of different types of pain, but Daniela hasn't fully examined GPR 160's role in all types of pain as yet, but we're getting there.
Subhashini Varadharajan says, fantastic presentation. Does GPR 160 have tissue-specific functions other than the ones you presented today?
Yes, absolutely it does. So my colleague, Joshua Stafford, who's one of my co-PIs, is actually investigating GPR 160's role in signaling in pancreatic islets. And so hopefully we'll have some really cool data to be able to share at Physiology Summit here in the future.
Carlos Perez da Costa thinks it was a very nice presentation and says it would be nice if Dr. Yosten added some bibliography for further consultation on this interesting topic. I believe, Dr. Yosten, that you have some review manuscripts out there that will contain a great deal of this literature.
Yes, that is correct. So, yes.
So they can PubMed search you.
Yes.
Bernard Ogola says, congratulations. The anorexia associated with GPR 160, is it gut or brain mediated?
We think brain, but there, of course, is definitely a possibility that there could be a gut component as well.
Patricia McDonald thinks it was a great talk and wants to know if 160 activity is regulated by beta-arrestin.
We think so. We think so, and we also think that there is, we think that there's a chance that the MAP kinase activity that we see when GPR 160 is activated is due to beta-arrestin mediated signaling. But I don't have the data to be able to say that for sure just yet.
Solange Abdul Noor Nakhul says, excellent presentation. Where is GPR 160 expressed in the GI tract?
It is definitely in the pancreas. I don't know about intestinal expression because we just haven't had the time to do a full IHC analysis of, a full body IHC analysis. I can tell you about expression of the mRNA, but again, that doesn't always necessarily tell you the whole story.
David Pollock wants to collaborate. He can send you tissues from their humanized sickle cell mice.
David, that would be great. Send them on. Let's do it.
Miguel Van Bemelen says, does GPR 160 heterodimerize, and could some of its functions depend on its association with other CRs?
Absolutely. I think its function absolutely depends on its associated with other GPCRs or other interacting proteins, such as integrins or ramps. We don't know what the identity of those are just yet, but it's such a common thing to see with GPCRs, homo or heterodimerization, that I would be very surprised if GPR 160 didn't do it too.
Okay, Fatan Sakaria says, thanks for the informative presentation. Is there any correlation between GPR 160 and CART with the brain analgesic system and secretion of endorphins or enkephalin?
We don't know. We don't know. It's a good question, but we don't know.
Well, I think we've hit the end of our time and the questions, actually quite a few questions for this one. Gina, thank you once again for this great presentation, and we look forward to presenting you with your award at the opening ceremony at the Physiology Summit next month in Long Beach.
Great. Thank you very much.
Bye, everybody.
Bye-bye.